Hello, and welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines. People just like you working to understand viruses and how they affect you. During the COVID-19 pandemic, we are talking with postdoctoral fellows involved in coronavirus and COVID-19 related research so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. With the promise of at least 2 highly effective vaccines for SARS-CoV-2, we still need to make it through the holiday season before these and other vaccines might be available. On November 16, 2020, we talked with Dr. Ben Israelow, an infectious diseases fellow in the Iwasaki lab at Yale, who has developed small animal models for SARS-CoV-2 and is using them to understand immune correlates of COVID-19 disease. Ben did his undergraduate studies at Washington University in St. Louis, and then his MD PhD degree at Mount Sinai School of Medicine, where he studied hepatitis C virus. He is currently at Yale in the Physician Scientist Research Program, where he has completed 2 years of internal medicine training and is in his 3rd year of infectious disease training. Hi, Ben. I'm happy to have you with us today. Why don't you tell us a little bit about yourself? How did you become interested in medicine and virology research?
Yeah, thanks for having me. I kind of got into that through my interest in infectious diseases. I went to Mount Sinai to pursue my infectious disease, to pursue my MD PhD program. At that time, I knew I wanted to do infectious diseases. At Mount Sinai, the microbiology department is mostly viruses, or all virology, and so it was, I realized that that's what I'd be studying, but quickly learned that viruses are fascinating, and the history of humans interacting with viruses really piqued my interest. How I got interested in infectious disease is a little longer. From a very young age, I've been very interested in science and started doing lab research as a high school student. I took a summer doing lab research. Actually, I went to Israel to the Technion Institute and did a summer research program doing molecular biology. Then, started, before I started undergrad, I joined a lab at Columbia University after I had already done some biologic research doing kind of more genetics research in diabetes. And so getting to WashU, when I started there, I knew I wanted to continue research. And so I found a lab actually over there at the medical campus doing neuronal stem cell differentiation. So embryonic stem cells were and differentiation and regenerative medicine was kind of new and looking very promising. And I was finding it super interesting. I transitioned into another lab while I was an undergrad doing more systems biology work in a plant biology research lab at the main campus. However, towards the end of my time at WashU, I was taking a class on kind of sociology of diseases, or the social impact of diseases, and one section of that was on HIV. And we read a lot of the research by Paul Farmer, as well as the biography about him, Mountains Beyond Mountains, and that totally clarified for me. I'd bounced around from a few different lab topics, but it certainly clarified for me that I wanted to do infectious disease research and wanted to put my efforts towards battling diseases that affected the whole world.
So I guess maybe a little bit more specifically, how did you identify the various places that you ended up? So the various labs or the various programs, how did you decide on those particular places?
Well, I guess once I got to WashU, I went to WashU for my undergrad because of the world-class biological sciences they have there. I was I grew up in New York area, in the New York suburbs areas. And looking, I was looking around the country. I wanted to leave this area and kind of explore a little more. Visited the university, and the resources that WashU puts towards sciences and et cetera is, they really prioritize it. And so I definitely knew I wanted to be at a place that prioritized what my goals were, which were biological sciences and research. And so I ended up there. When I was in high school, I was interested in neuroscience, and so that's how I got into that first kind of embryonic stem cell lab, or a neuroscience lab, differentiating neurons. I was there for 2 years. I learned a ton about differentiation and those, and as well as like patch clamping and neuron techniques and stuff like that. During that time, there was, I guess, microarrays were starting to become more popular, and so we were doing microarray work with some of our differentiating neurons under like different conditions. There was a summer research program at WashU through the NSF called Fiber, which was like Frontiers in Integrative Systems Biology or something along those lines. And so it was a summer program where I'd be taking classes and doing research in systems biology, so learning how to interpret kind of more large scale data sets. And I thought, I could apply that to the neuron differentiation work that I was doing, and B, this is like a great opportunity to both collaborate with people I wouldn't normally, so people in statistics and mathematical modeling, et cetera, and learn these new skills of kind of more systems approaches to biology. I ended up staying in that lab because I enjoyed the work and found it interesting, and it kind of gave more avenues to continue this kind of exploration of systems biology. I guess I kind of mentioned how I went from there into kind of infectious disease, but for a while I was deciding between a PhD program or an MD PhD program. I kind of came to it more from research, and I was definitely motivated or enticed or intrigued into medicine from what I learned about the history of HIV and as well as infectious diseases.
And then how did you get into your current lab? Or how do you get into your postdoc lab?
At Mount Sinai, I knew I did my PhD research on viruses, on hepatitis C. The transition from your PhD to back to MD, the last phase of your MD during an MD PhD program is when you get into the clinical world. And I kind of went into that with an open mind, saying like, I think I'm interested in infectious disease, at least from a research perspective, but let's see what the medicine's about. And, from rotation to rotation, I continue to be like, oh, I really like the infectious disease aspect. This is what I'm interested in. So I continued with that, and I joined Yale's physician scientist track, which is I guess they call it a short track, where you reduce your internal medicine time from 3 years to 2 years. And then they add an extra year of research onto so it's still 6 years, like a 3+3, but it's more like a 2+4. And so I came to Yale because I had been doing virology, and I knew I wanted to kind of expand that more into immunology, and the immunobiology department here is world class and very impressive. I wanted to do viral immunology, and so that naturally led me to the lab of Akiko Osaki. Not just in terms of the work that she does, but in talking to people around here, and everyone I talked to was like, you need to join Akiko's lab. She's doing amazing stuff, and especially for me, who's wanting to branch out from being more molecular virologist to increasing my my skills and my knowledge of immunology, and to be able to kind of more apply them was clear.
Great. So can you, I guess, tell us a little bit about the work that you've been doing in her lab? So either, I guess, pre-COVID maybe, and then and more recently on COVID-related research.
Yeah, absolutely. So I joined Akiko's lab in August, before the pandemic, and so I was not there for that long before the pandemic hit, but I joined to kind of continue doing some viral pathogenesis work. So part of my PhD on hepatitis C, I was looking at how different interferons are induced by the virus. And so I wanted to continue that, but into animal models, and looking at how the role that interferons play in pathogenesis of viral infections. They obviously play an important role in preventing viral infections, but they also play a role in pathogenesis. And so some people in the lab in the years before I got joined had been doing some Zika work, looking at interferon pathogenesis in the perinatal period, and so pathogenesis towards the fetus more than I guess the other host, the mother. And so I wanted to kind of continue some of that work, and looking at what the role of like different interferons play in healthy or not healthy pregnancies. And so had started with that, and I hadn't really done any mouse work before, or had done minimal flow cytometry, and so this was definitely a learning experience, especially also having been out of research for 5 years, being back in the clinical world. And so during that time, I was doing experiments, learning, and in January, when we first started to see this happening, Kiko and I had a discussion about, should we start investigating, maybe doing some research here. Akiko, I guess, had difficulty doing research on highly pathogenic viruses in the past, in terms of what our environmental health and safety allowed. She'd tried doing some H5N1 work in the past that never was allowed to happen, and so she was a little hesitant about jumping in, and was like, if you're interested, we can start this. And so I started investigating this and what to do, and it became clear, especially about how to research this, especially considering the tools that the Iwasaki lab had in the mice colony, and that we needed to be able to study this virus in a mouse. And so the first projects that we started on were developing a mouse model. We took a few roads towards that, including viral adaptation, as well as the paper that we published on using an adeno-associated viral vector to be able to promote viral infection in mice of different strains. And so that's kind of how it led me towards developing a mouse model, and furthermore studying the role of interferon in that. What we've been getting more into recently is trying to we've done a lot with the pathogenesis of the virus, both in mice as well as in humans. So once we started having a lot of cases in Connecticut in April and May, Akiko and a bunch of other researchers here in public health and other departments kind of got together and developed a biorepository for coronavirus patients, mostly hospitalized coronavirus patient samples. And so there was a very talented postdoc in our lab who's kind of leading that. And I was helping them doing, data extraction from charts and getting that information. So, between the human coronavirus SARS-CoV-2 work and the mouse stuff, we really saw that a lot of the questions now are beginning to revolve around what constitutes a successful, clearance of the virus, as well as adaptive immunity to the virus. So how, it's from early studies, it seemed clear that most infected people developed adaptive immunity to the virus, but not everyone had successful outcomes. And so the questions of that we're starting to address are, what constitutes a successful adaptive immune response, and then furthermore, what controls the adaptive immune response. And so those are some of the questions we're starting to try to answer right now. There's a lot of SARS-CoV-2 research going on in the lab right now.
So I guess I'm thinking more broadly, what was the most exciting science moment in your career so far? So it doesn't necessarily have to be earth-shattering, but something that was exciting to you.
I think it's usually when you get unexpected results, is what I've found. And so, so part of the initial part of my work on hepatitis C virus was looking at different host factors that were required for hepatitis C virus replication. And one of them is so hepatitis C is very liver specific, human liver specific, and part of the cell tropism are some of the entry receptors that are liver specific, but another one is a microRNA called miR-122. It's very conserved from like zebrafish through humans. And the virus is adapted to utilize this microRNA, not by the microRNA physically binding to the viral RNA. And it looks like it acts to protect it, but may also act as a translation replication switch and a few other things. And so we were working on this. An MD PhD friend of mine had been developing a microRNA screen where he'd built a library of microRNA decoys and needed to kind of validate that. And I was like, oh, we could do that with hepatitis C. It'll kill all the cells except the microRNA miR-122 decoy expressing cells. And so we did this. We got some hits that were interesting for, cell survival, et cetera. Nothing too earth shattering, but in our control cells, which were the population of miR-122 expressing cells, we started to see that they started to get infected by the virus, and which was very unexpected. And so we sequenced we were like, this can't be true. So we repeated it. And again, the virus, after, passaging the virus on these cells, it infected them. And we sequenced the virus, and sure enough, like the virus had developed mutations to allow it to adapt to be able to replicate, start almost in the absence of microRNA miR-122, which, was completely surprising. And, working towards figuring that out and doing experiments and figuring out what that meant was really, really important. I think it was cool because it was a project where, I was collaborating with, a fellow student that we were friends with, and just the whole collaborative process was great.
Cool. So I guess on the converse side, what's been the most difficult thing you've had to overcome as a scientist, and how have you overcome it?
It's always difficult as an MD PhD, it's difficult to transition back and forth. When you're during your PhD, you defend your thesis, you're the smartest person in your specific field, and then you go back into the clinic where you're a medical student and know less than all the other medical students because you've been at it for a while. And so that's always difficult. You learn to become humble very quickly. And then again, coming back here and joining the lab, you're like, I did know how to do research at one point. And so there's a lot of questioning yourself about, or at least for me, there's been a lot of questioning, was I just lucky during my PhD? Do I know how to do this? I guess a lot of imposter syndrome that comes up. But you kind of just keep going and pretend that it doesn't exist. I don't know.
I mean, are there specific things that you do to sort of overcome that sort of transition back and forth? Is there strategies that you use?
I just try to remind myself that, you've made these major transitions before, and for some reason or another, they turned out to be successful, and let's just cross our fingers and keep going. And hope that they'll be successful. I have a supportive partner, and now I have a 2-year-old son, and so while the transitions, you could imagine being harder with them, they having them at home and having a support system, I think is really important, and being able to be like, come home and have my son say amazing random things to me kind of puts things into perspective.
Yeah. So I guess more personally, how has the COVID-19 pandemic affected you as an individual? And then I guess since you're a physician, do you have any sort of insight into how it's affected, life as a doctor?
Yeah. So I get, I was paying very close attention in January, and then in February, as the outbreak in Italy was happening, I was getting increasingly concerned that this was not going to be isolated in any respect. And so I think in the last week of January, I wrote an email to my family and friends saying, like, everybody, you need to get ready for this. And so for the beginning of the pandemic, I'm an infectious disease doctor, but I'm also a partner, a husband, a son, a nephew, a cousin, etc., friend. And so, while I've been a doctor for a lot of people, I'd never been a doctor for so many that I was related to, all of a sudden. And so that was a huge change and very striking, as the uncertainty of this was clear and profound. People just didn't know what to do. People are calling, asking, oh, can I, like, things benign, like very simple things, like, can I go out to the store? Like, should I, all those questions that people were addressing. Like, I was getting those calls. I was also getting those calls because at Yale, they set up a call center and wanted infectious disease physicians staffing it. And so as a research fellow who wasn't in the hospital 100% of the time, we were helping to staff that. And so getting those questions from doctors and everything, and the questions from doctors were different. They were like, how should I structure my office? How should, do I need to cancel all my visits right now? And those decisions were changing from day to day, week to week. And so it was something, you don't get trained for and you don't expect to have to deal with. So, it was certainly interesting and challenging to have to be dealing with that on the one hand, and also trying to get research moving as fast as we could as the virus was coming and the university was shutting down and all that. So yeah, in terms of how as a doctor, I think it's been hard for a lot of us, especially now, even harder, because we're seeing the public health maneuvers that could have helped prevent these secondary and tertiary outbreaks that are, we're seeing a huge spike again in Connecticut. We didn't, I think Connecticut slowly opened and they were good and we didn't have a second spike, or this is now our second spike, but we're the hospital, we have about half as many hospitalized COVID patients currently as we did during the peak in April. And that's only going up every day. It's just I think it's very disheartening. And, I heard somebody mentioning this on, like, in April and May, we were the, support our heroes, et cetera, et cetera. That's just not the narrative right now. So, for whatever reason, for better or for worse. And so I think it's difficult to have to go through this again. Yeah, a little post-traumatic.
Yeah. How do you, so I guess, how do you take care of yourself sort of mentally, not, to get through this?
Yeah, I've been trying. I've been trying to more prioritize, getting family time on like weekends, and not being in the lab 24/7, especially now after there was that intense rush during the first 6 or so months. um, Try to spend time with my wife and my son, and really decompress and leave the lab for a little bit, because we're going to be in this for a little while longer, and it's it's a race, but it's a marathon, and so keeping
Yeah.
keeping your endurance is important. You don't want to tap out at mile 12.
Right. Yeah. So I guess sort of to follow up on sort of safety issues, as a virologist and infectious disease doctor, how do you make decisions about how to keep yourself and your family safe? So how do you think about that risk?
Yeah, I mean, I try to stay on top of cases, because the risk changes daily, and it really depends on what's going on in your area. I mean, I definitely recommend to people listening, because I get calls from friends in different parts of the country. I'm not always up to date. I say, really listen to your public health people. They're the ones who are staying on top of this. They're the ones who, just follow their recommendations. For me, I am also following those recommendations. Trying to, it's always a balance about safety and versus living your life. Like, my son is currently still in daycare, but our daycare is very small, and so we're weighing those risks currently, and talking to other people whose children are in school about weighing those risks about, should we be thinking about taking them out soon, etc. And so again, for me, I just try to stay on top of what's going on in the community. That's the major indicator of risk. It's interesting, because during the first wave in April and May, we didn't have testing, and so the numbers now definitely look a lot worse. But I think what's more indicative of what's going on is the hospitalization rate, and so it's clear that this is totally out of control again in our area, and so I think we're paying attention. For me, we've decided that we're just not going to be celebrating Thanksgiving with other people this year, because it just doesn't make sense at this point in the pandemic, where we are, where Connecticut is, in terms of how bad it is. And we're still 2 weeks away from Thanksgiving. It's only going to, I can only see it getting worse currently, and so we may try to see people outside, but it's too cold to really have a Thanksgiving meal outside, at least at this point.
All right, well, great. We're wrapping up a little bit. Any last messages for our listeners as we're heading into Thanksgiving? There's now good reports that we may potentially have 2 vaccines, but obviously not until April, maybe next summer for the majority of people. So any thoughts about the future of the pandemic? Where do you see us going?
Yeah, I definitely think the vaccine data is super, super encouraging. I think based on the fact of how quickly we were trying to get vaccines out, I would have been happy if it was like 50 or 60% effective. Seeing 90% results is very, very encouraging, especially to get this under control and get our lives back to normal. I think we're going to be dealing with SARS-CoV-2 for a long time. Questions I think still remain about the durability of protection of these vaccines. I think obviously questions still remain about, like, will this data hold in larger groups? But I think at least the starting point of 95% is really, really promising. And so I think we're going to be, until we get those vaccines on a more, systemic, wide scale to be available for people, the public health measures are what's gonna, keep us safe. And so we're obviously wearing masks, avoiding, again, depending on where you are, avoiding maybe even small social gatherings. And so that's, I think, where we are right now in Connecticut. I wouldn't have said that 2 months ago. I would have said, small social gatherings are, great, whatever, perfectly acceptable. But right now, where we have just rampant widespread, of the virus, and there's no like clear super spreader events that we're tracking them to. They're all small gatherings of people, and when it is this spread, we have to kind of reduce transmission rates, and the way to do that is, stopping contact.
Great, thanks for talking with us today. Ben is currently using small animal models that he developed and human patient samples to understand what contributes to a good outcome and protective immunity following SARS-CoV-2 infection. This has been Let's Meet the Virologists, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, Spotify, and other podcasts, or at lmtv.podbean.com. If you are a virologist interested in sharing who you are and what you do, please contact us at letusmeetthevirologists@gmail.com.